Hello everyone, my name is Abiduraman. I belong to Department of Plant Biosciences and I'm also a member of Agri Innovation Center. Today, I'm going to introduce one of our research topics, identifying the molecular regulators in quest of developing cold stress resilient plants. So let's look at some of the facts about cold stress and its impact on agriculture. In 2009, the low temperature alone caused crop damage worth 158 billion yen in Japan. Then also early and late frosts damaged the production of vegetables and fruits worth 5 to 6 billion yen each year in Japan. And the more alarming thing is, because of the climate change, it has been predicted that more snow will fall in the interior land of Japan when no snow was encountered before. So based on that, it is imperative for us that we should think about developing cold stress resistant plants. Now, you all know that cold stress inhibits the plant growth, and this is an example where you can see at optimal temperature plants grow very nicely, but at cold stress, it doesn't grow much. And previously we showed that this is linked with plant hormone, very important plant hormone called auxin, and these blue stainings represent the cellular auxin signaling. And you can see here at 4 degrees Celsius, when we can see the accumulation of auxins in the root meristem zone. That tells us that cold stress alters the intracellular auxin homeostasis. Now, to understand the pathway of auxin, intracellular pathway of auxin, we need to understand the auxin transport system. So auxin is synthesized in the meristematic tissue, particularly we call it shoot apical meristem, and also in the root apical meristem. And then this auxin, once it is synthesized in the young tissue, then it goes downwards from the top. And on the other hand, at the bottom, when it's produced the root tip, it goes towards the shoot. So we call that there are two transport systems working for auxin. One is rootward transport and the other one is shootward transport. So this is rootward and this is shootward. And it's very important to understand that this has a polarity. That means it has a directional movement from bottom to top, from top to bottom. And for this, actually auxin, for transport of the auxin transport proteins are required. Inside the cell, there are different family of proteins which actually transport the auxins. One is the auxin influx carriers. That means that for the intake of auxin inside the cell. And the other one is the efflux of auxin, which means from one cell, it passes to the other cell. And it is pins and ABCBs function in this. So as I told you here that this is a polar transport of auxin, so we expect that the proteins will be localized at one end of the cell, and you can see exactly what is happening here. These are localized at the one end of the cell. And more interestingly, another interesting thing is that these proteins actually cycle from the membrane to cytosol and again come back to the membrane. So this is we call the protein trafficking. So basically there are two things going on. One is the polar localization of the proteins, and the second one is the trafficking of the proteins. So when we treated with plants with cold stress, we can see there is a huge reduction in the auxin transport. So you can see here 50% reduction. And as I told you, there are two possibilities. One is the polar localization of the proteins or the protein trafficking. And you can see here when we treated the plants with 4 degrees Celsius, we don't see any effect on the polar localization of the proteins. They are perfectly all right. But if you look at here, this is we call the trafficking. It's coming from the membrane to the cytosol and going back. This trafficking is completely blocked at 4 degrees Celsius. So that tells us that the protein trafficking is a very important thing for cold stress response. So inside the plant cell, there are three pathways for protein trafficking. One is the recycled endosome, then the other one is the late endosome, and the other one is the early endosome. So there are many proteins. We know there are many proteins working on this. So because we thought that this would be important, probably the protein trafficking pathway is important for cold stress response, what we did, we screened all the mutants that are present in this pathway for cold stress response. And we actually screened like 40 mutants with single and double mutants. And you can see here, we found only one mutant that is called NOM-BE, is hypersensitive to cold at all time points that we tested. And if you go back, this NOM is actually functioning in this recycle endosome pathway. So because NOM is hypersensitive, so we thought that this could be a direct target of cold stress, and we found that that is indeed true. When we treated the plants with expressing the NOM GFP at cold stress, you can see the signal goes down, GFP signal goes down. That means that cold stress is inhibiting the NOM expression, but it comes back when we put them back in the 23 degrees Celsius. So based on this, we make a hypothesis that, okay, if when there is no NOM, it becomes sensitive to cold stress. So if we overexpress NOM, that it will confer cold stress resistance to plants. So we generated overexpression lines. You can see here, line 1, line 2, line 3. All of these are produce 10-fold more protein compared with the control. And then we checked their cold stress resistance, and indeed we found that that is true, that NOM overexpression lines are actually resistant to cold stress. And then we also looked at the whole plant level. This is the early seedling stage. You can see these are very tiny plants. And then we looked at for the grow them in the cold, continuous 40 degrees Celsius for 6 weeks. You can see here, and you can see this is the control Columbia plant. It grows a little bit, and the mutant is very sensitive, and this overexpression line grow very, very well. And after 8 weeks, you can see this is the result. This is almost dying, the mutant, which is very hypersensitive. The wild type, it didn't die, but it grows a little bit only. On the other hand, our normal overexpression line grow very well. It even flowers. So based on this, we can understand this NOM protein is very important for plant cold resistance. So what is the future perspective? One important thing that we have to remember that NOM is a highly conserved protein across the plant species. So we think that cold resistance in different crops can be increased by manipulating only the single protein. Actually, we are working on this with the NOM protein in tomato and rice, and we hope that we'll be able to develop cold tolerant rice and tomato plants very soon. Thank you for listening.